good evening. Welcome to Aakash BYJU'S NEET channel. I'm your teacher Ankita and I welcome you all in today's class. I hope that all of you are doing amazingly well and I hope that all of you are ready for a very interesting class where we will be discussing about the search for the genetic material. In short, we will be looking at the experiments that happened and that actually helped in scientists concluding that DNA is the genetic material. So I hope everyone, all of you are ready with your notebook, with your book, with your pencil or a pen, because today's class will be a super interesting class, everyone. Yes, good evening everyone. Good evening to each one of you. Good evening Naruto, Mayuk, Tanu, Bhumi, Neha. Good evening everyone. I hope that all of you are doing really well and your health is super awesome and you are excited for the class today. Awesome everyone. Amazing. So now in today's class we know that what we'll be discussing. It could be that you're watching this video for a very first time. It could be that you're a new subscriber on our channel. So don't worry, this is the lesson 3 or the part 3 of the chapter The Molecular Basis of Inheritance. We have done 2 previous videos. First one was on the structure of the DNA. We got into the detail about the structure of the DNA, the whole structure, and then of course the next class we discussed about the important characteristic features of DNA and the packaging of DNA. So we have L1 over here and L2 and today is the part 3 or the lesson 3. So I hope that everyone, all of you are ready for the class. Awesome. So let's get started. Hello Sanchita. Welcome. Good evening everyone. Welcome. Yes, very good. That's good. So let's get started everyone. But before we get started, let me quickly call out. In the last class we had a discussion about this question and I gave you as a homework. So I saw that many of you have replied, right? It was a good thing, right? It's just a very good thing, when of course we follow through what is there. Because what happens, there are times when we really want to, we take up a class, we go to the questions, and of course we do what we can do. So here everyone, let me quickly call out the names of the students who actually have written the answers. Just calling out name everyone. Probably the next time I'll have your answers also on the screen so we can see our answers. Yes. Hello Shalini. Okay, so we have answer given by Naruto, then we have Aditya, Shalini, Mayuk, Abhishek and Life Science have given us a detail but not the answer. But yeah, still great. Okay everyone, so congratulations. Thank you so much everyone for answering over there and you are absolutely correct with the answer. So let's quickly take a look at the answer, right? We talked about the percentage of the cytosine is in a double stranded DNA 17. What is the percentage of the adenine in DNA? Of course, many of you have answered. Let's see the solution for it. So we know that cytosine, we have how many percentage? 17%. We know that A plus T is equal to G plus C and of course A is equal to T. If A is 120, T will be 120. So in this particular case, C and G will be 17%. Then of course we will be doing the total of it and of course we should have 100%. So we have G plus C ka total, which is 17 plus 70, that is 34. 34 we have over here. If we move this 34 over here, we have to minus 100 minus 34, we have 66. 66 divided by 2, that gives us 33%. So the answer was 33. Option number C was the correct answer. Now the other question was, the segment of DNA has 120 adenine and 120 cytosine bases. We have to find the total number of the nucleotides. Of course we know that it will be same, right? For of course whatever be the A will be the T and whatever will be the G will have the same as C. So if we have this 120, this 120, 120, of course we have 4 nitrogenous bases. The total answer is 480. So that is option number D. Very good everyone. So I hope that all of you got this question. Good. Very good. Right? I hope that all of you got this question. Quick thumbs up everyone. Quick thumbs up. Interesting. Very good. So now that we are clear with this everyone, let's get started. Now in today's class, we will be discussing about the different experiments. Before that, let's quickly take a look at a very short history that we have. Hello. Yes. Arsina. Hello Arsina. Chaliye. Let's see everyone. So we know that in a previous class we discussed about Misha, right? Anyone can tell me what he did? Yes. Oh, that's nice. Vitamin proteins. Nice. Yes, everyone? Everyone. Right? Mishra, right? We studied it about him in the last class. He played a very important role. He did a very interesting experiment in the year 1869 and he actually found about the nuclei, right? Nuclein. You remember this? Yes, and from there, of course, the beginning started, the search of the genetic material started. So, from 1869, of course, we can see over here, right? Years went by, we have Mendel's experiment, and of course, we have various scientists who have contributed. Then, right, we have Morgan also, I'm sure you have studied about this in the previous chapter. So it took us a while to understand, right? We knew that we have some genetic material. We had a hint about it. We were looking for it. We had the clarity that there is something that is actually helping in the transformation. Not the transformation, but from one generation to the next generation, it's being passed. Ye, we were all clear about it, right? ये हमें थोड़ा hint मिल चुका था. Yes? Yes, this is for NEET 2024. Absolutely correct. Bilkul. NEET, whatever we are discussing over here is based upon the NCERT and NCERT is definitely enough for the examination. Okay? So now everyone, what we will do? We will be moving ahead and we will be discussing about the experiment. But before that, everyone, in our textbook, they have mentioned about these scientists' names. I would love all of you to take a screenshot of it, bachcho. Jaldi se take a screenshot of this. Really sorry for the delay. We had some issues with the studio. That's why we started our class at 7:15. Yes. Okay. Everyone take the screenshot so that you can come back and read about them. Okay, what are the contribution of these scientists, right? Chaliye. Now we'll be discussing about the Griffiths experiment. What did the Griffiths did, right? Of course, there's a very important experiment that we have in Griffith ने क्या करा उस experiment में हम लोग वो देखते हैं. चलिए. Right? Griffiths experiment. Now what he did was very interesting, right? Yes? Very good, right? What he did, hello everyone. Let's just focus everyone, yes. Okay, now everyone, can you quickly tell me I'm sure that you have read the chapter and you're coming to the class. If not everyone, let's make a point that when you're coming to the class, you're just going through the textbook once so that you have an understanding that what it is saying. And over here, we can come and discuss about doubts and we can learn in an amazing way. So we know that he did an experiment with the Streptococcus pneumonia, right? All of you, very good. So we have two strains of it, right? We have two strains of it. We have R strain and we have S strain. R strain is pretty much rough. S strain is very smooth, and because it is smooth, it can actually invade into the immune system of the other organisms and can cause some damage. Whereas the R does not have, right, a coat. So of course it can be easily detected by the immune system of the host and it will not be able to cause any harm to the host, right? It will not be able to cause a disease. So we have two strains of it, right? We have two strain, R strain and S strain. So what Griffith did is very, very interesting. He took this, right, S strain. Right, everyone? He took the S strain and this S strain was virulent. Basically, it was an active one. Yes, it was an active one. Active. Yes, it can cause the harm. And of course, this does not, it's not very active, right? Basically, it does not have that many activities in that such that it can cause a disease. So, the one that will be causing the disease, we call it as the virulent, right? Clear? So then he added this S strain into the mice. And can you guess what will happen to this mice? Jaldi say. Very good, Naruto. Very good. Yes, welcome Tamanna, Namrata, Saloni, Kabir. Very good. Yes, we know that the mice will not be able to survive and it will die. Mice will say bye-bye. I'm really sorry, I cannot take it more, and of course we will see that. Unfortunately, we see that mice will not be able to survive. Then what he did in one more experiment in a sense. He added the R strain to the mice, and what he saw, the mice lived happily ever after, right? Yes? Very good. Now you know what he did? He added the S, heat killed strain. Right? He added the heat killed strain, and what he saw, that mice will live. Yes? Nothing will happen and mice will live. Then everyone, what he did was very interesting. He added the S strain ka heat killed strain plus R strain. The R, which were very innocent, right? R strain is very very innocent. Ekdam pavam sa hai ye. Right? So R strain is very sweet, is not doing anything. So when he combined the S strain, which was heat killed, with the R strain, which was very innocent, something very drastic happened. What happened, everyone? We saw that mice could not survive. Yes, everyone? Yes? All of you, are we clear? We saw that mice could not survive. तो ये तो बहुत easy हो गया ना? Everyone, are we clear with this? क्या हुआ इस experiment में? हमने देखा कि हमारे जो साइंटिस्ट थे ग्रिफिथ, उन्होंने क्या किया, एक एक्सपेरिमेंट किया. किस पे एक्सपेरिमेंट किया उन्होंने? दे यूज दी स्ट्रेप्टोकोकस निमोनिया, राइट. उसके दो स्ट्रेन होते हैं. वी हैव आर स्ट्रेन जो रफ है, उसके ऊपर पॉलीसेक्राइड की कोट नहीं है, वेरएज एस जो एस स्ट्रेन है उस पे कोट है, म्यूकस पॉलीसेक्राइड की कोट है. सो जब एस स्ट्रेन है दैट कैन इजीली right invade into the immune system and can cause disease, whereas the R right which is rough strain cannot cause disease and it can be easily killed by the host immune system. इसके पास में कोट नहीं है, इसके पास में कोट है. तो ग्रिफिथ ने क्या किया, कुछ समझने के लिए कि क्या है, क्या मांझरा क्या है, कि क्या हो रहा है, उन्होंने 4 सेट में एक्सपेरिमेंट्स करे. पहले क्या किया उन्होंने, एक एस स्ट्रेन लिया जो फुली एक्टिव था, जो पैथोजेनिक मतलब जो एकदम ही एक्टिव है और जो डिजीज कॉज कर देगा, उसे माइस के अंदर डाला और माइस मर गया. दूसरा क्या किया, आर स्ट्रेन डाला तो ऑफ कोर्स आर्स का कोई फर्क नहीं पड़ा आर स्ट्रेन का, और माइस खुश रहा आराम से कि हाँ मैं तो जिंदा हूँ, कोई दिक्कत नहीं है. फिर एस स्ट्रेन को हीट किल किया, मतलब गर्मी से ऑफ कोर्स इसको बैक्टीरिया को इनएक्टिव कर दिया, मार दिया बेसिकली. तो क्या हुआ, माइस हमेशा फिर से हैप्पी है, जिंदा है. फिर उन्होंने जो लास्ट करा उसमें क्या किया उन्होंने, एस स्ट्रेन का जो हीट किल्ड स्ट्रेन था उसे प्लस आर स्ट्रेन जो एकदम इनोसेंट था, जिसने अभी तक किसी भी चूहे को नहीं मारा था, उनको साथ में मिक्स कर दिया और चूहे के अंदर डाला. तो क्या हुआ, माइस सर्वाइव नहीं कर पाया. एवरीवन, आर वी क्लियर विद द एक्सपेरिमेंट? ऑल ऑफ यू क्विकली गिव मी अ थम्स अप, जल्दी से. हाँ, नारुतो विल डिस्कस, अभी वी विल डिस्कस. भास्कर, क्योंकि ऑफ कोर्स रिएक्शन हुआ, राइट? वेरी गुड, क्लियर. सो दिस इज क्लियर, राइट? सो अब हम देखते हैं व्हाट वाज द कंक्लूजन ऑफ इट. दिस इज एक्सपेरिमेंट एंड फिर ग्रिफिथ ने ही कंक्लूडेड दैट कि देयर इज समथिंग दैट इज बीइंग ट्रांसफर्ड, राइट? कुछ ना कुछ ऐसा हुआ है जो ट्रांसफॉर्म कर रहा है. Clear? There's something which has been transforming the S strain. So he just left it that. उन्होंने बोला transforming principle. Clear everyone? अब इसको with the help of some slides पढ़ते हैं. I want all of you to pay attention now. See what we have everyone over here. You can take the screenshots along with me, right? So we have the Griffith. उन्होंने Streptococcus pneumonia जो बैक्टीरिया है जो कॉज करता है निमोनिया, वो लिया, उसके 2 स्ट्रेन लिए, कल्चर किया, उन्हें प्लेट पे ग्रो करा. 2 स्ट्रेन थे, कौन से था, एस स्ट्रेन था, जो जिनकी कॉलोनीज कैसी थी, स्मूथ शाइनी कॉलोनीज थी. एंड ऑफ कोर्स वी हैव द रफ कॉलोनीज विच आर आर. फिर उन्होंने क्या किया, एस स्ट्रेन पे उन्होंने बोला कि वी हैव द म्यूकस पॉलीसेक्राइड कोट. दिस इज इंपॉर्टेंट एवरीवन, आई एम मेकिंग अ star over here, please note this down. S strain पे क्या था? They have the coating of mucus polysaccharide. Basically उनके पास में क्या था? Polysaccharide का coat था. Clear? Yes. Then we have the R strain which does not have a coat. This coat actually helps the bacteria to invade the immune system, and of course it can easily infect the host animal. Yes, S strain has a smooth coat, right? Smooth bacteria, and thus it's virulent that can cause diseases, whereas R strain is not. Clear? तो ये हमें पता है. Here everyone, what we have? See, we have S strain and we have R strain. We can clearly see यहाँ पे कोट दिख रहा है, polysaccharide coat is there in the S strain, whereas R पे हमारा polysaccharide का कोट नहीं है. Clear? तो हमें पता है कि कौन सा एक्टिव है, कौन सा इनएक्टिव है. उसके बाद में हमें पता है कि क्या होगा. एक्सपेरिमेंट करा हमने चूहे के अंदर, राइट? हमने नहीं करा, ग्रिफिथ ने करा, और उन्होंने क्या किया? वी हैव द लाइव स्ट्रेन एस, ठीक है, लाइव स्ट्रेन लिया एस जिसके पास में पॉलीसेक्राइड कोट है माइस के अंदर, एंड माइस सी ओवर हियर, माइस इज नो मोर, राइट? यहां पे हमें दिख रहा है. देन ऑफ कोर्स वी हैव आर स्ट्रेन. आर स्ट्रेन में क्या हुआ? माइस इज रियली वेरी हैप्पी. Then the third step comes everyone, right? Heat killed S strain. Heat killed S strain को जब माइस में डाला तो क्या हुआ? No living bacteria was isolated. कोई भी living bacteria हमने isolate नहीं किया माइस में से, माइस जिंदा था. The last one was a shock to the scientist that heat killed S strain और live R strain को जब साथ में लाया तो क्या हुआ? एस स्ट्रेन बैक्टीरिया आइसोलेट हुआ डेड माइस के अंदर से. तो इवन दो यहाँ पे जो एस स्ट्रेन हम सोच रहे कि मर गया है, राइट, वो माइस के अंदर में से उसके सेल्स में से आइसोलेट किया गया. सो देन बेस्ड अपॉन ऑल ऑफ दिस व्हाट ही कंक्लूडेड दैट देयर इज समथिंग जिसने आर स्ट्रेन को ट्रांसफॉर्म कर दिया एस स्ट्रेन में. वो समथिंग was not clear to him, and what he called it as? He called it as the transforming principle. Everyone, are we clear? Everyone, are we clear? Quick thumbs up, everyone. I know that. I'm sure कि आज पूरा दिन आप लोगों ने पढ़ाई करी, थोड़ी सी एनर्जी हो सकती है थोड़ी सी कम हो, बट ये फर्स्ट एक्सपेरिमेंट हमें समझ में आया. देखो, एक्सपेरिमेंट वैसे बहुत ईजी है, इट्स अ वेरी वेरी ईजी एक्सपेरिमेंट. क्वेश्चंस बहुत सारे आते हैं, है ना, इसमें से. तो हम हर माइन्यूट डिटेल पे फोकस करेंगे जो हमारी एनसीईआरटी में है. समझ में आ गया ना सबको? तो क्या हुआ? आर स्ट्रेन समहाउ एस स्ट्रेन में कन्वर्ट हो गया. कोई जादू हुआ और हमने देखा उनके हिसाब से कि आर स्ट्रेन एस स्ट्रेन में कन्वर्ट हो गया, बट उनको नहीं पता था कि व्हाट इज दिस. सो ही कॉल्ड इट एज द ट्रांसफॉर्मिंग प्रिंसिपल. ओके, एंड he said that कि R strain from the heat killed S strain, मतलब there is some transformation happening. So the transformation principle enable the R strain to synthesize something. He said that कि R strain actually got something, right, so that it can synthesize smooth polysaccharide coat and become virulent, right? So he said that कि कुछ ना कुछ हुआ, S ने कुछ R को दे दिया ताकि R के पास में भी एक स्मूथ कोट आ जाए और उसकी वजह से वो भी एक्टिव हो जाए. Yes, I will try. SJ, I am trying. So what is happening over here? Majority of the class will be there in the English only. Don't worry. When I'm repeating the concept, I might repeat in Hindi. Okay? So don't worry. I know that we all will be writing our examination in English, and we really want to keep English as the medium over here in terms of teaching. But I know that there are so many of you who are here who wants me to repeat the concept in Hindi at least once. So I'll do that. Okay, so it's a win-win situation for all of you. Clear? We'll do that, right? It's a win-win situation for all of us, right? Hindi mein bhi hai, and we have it in English also. Win-win situation for all of us. Quick thumbs up, everyone. Quick thumbs up. Yes, hello Deepu. Very good, very good. Good to see that all of you are here and energy is super high. Geeta Ben, yes. बच्चे, I am not sure it's purely based upon your interest, that what you want to pursue. Please talk to your parents, please talk to your seniors, right? And I'm sure they'll be able to help you with that. Yes, chaliye. Okay. Now everyone, here's a question time. We're done with the first experiment, everyone. Pat on our back. Pat on our back. Let's move ahead. Which of the following result is not consistent with Griffith's experiment? It's a very tricky question. Alright? It's a very tricky question. Which is not? Everyone pick the correct answer. See, he was following something, right? He was doing an experiment and he thought that, aisa hoga to aisa hoga, aisa hoga to aisa hoga. But he got a shock. He got a shock at the last experiment. That was not in the regular way it should be happening. Yes? Read the question carefully, everyone. I'm giving you kind of a hint over here. It's a tricky question. The question is asking which of the following result is not consistent. Consistent means बार-बार होता रहता है ना? With the Griffith experiment, but very good, everyone. Option number D is the correct answer. S strain डाला, तो of course we saw that mice died. R strain, mice was living. Then injected heat killed R strain. What happened? Of course, we saw that S strain, when heat killed, the mice was living. This is a condition which was a shocker for him, right? It was a shock thing for him also. So option number D is the correct answer. Clear? So here everyone, take a screenshot of this jaldi se. Quickly everyone, it's a very quick revision for all of us to have. See, strain type, right? We have the virulence, we have the transformation, and we have the result. Okay? Very good everyone. Quickly, take a screenshot. Yes, and I hope that all of you are enjoying, everyone. Yes. Okay. This experiment happened in which year? It happened in the year 1928. Remember this. I know. Yes, it happens. See, question can be tricky. The concept is same, right? But of course, the examiner can ask you question in a very tricky way. Chaliye. So here's everyone, Kaju Katli for all of us. We'll have laddus also today. A change in the sweets that we have every day. Okay. One more question, everyone. Please do read the question carefully. And here we have, which of the following correctly describe the difference between S, R and S strain of Streptococcus pneumonia, right? Bacterium in the Frederick Griffith experiment. What is a difference between R strain and the S strain? Yes, very good. I can see one correct answer, two correct answer. Now, of course, I can see a lot of correct answer. Very good, everyone. The difference between the S strain and the R strain is about the coat, mucus polysaccharide coat, right? While R strain does not have it. So, option number B is the correct answer. Very good. With this, everyone, we are done with the very first topic that we have today. That is the transforming principle. Please, everyone, this is your NCERT. Please do read it. I will have the snips, right? Please make sure after the class gets over, today only just go through it once. We have the important here. We have the name of the organism. Super important. We have the S strain and R strain. What are these? What is the covering that they have, right? Then please do mention about the organism on which the experiment was happening. It is mice. We have two types. We have active and inactive type. We have all of this, right? And of course, we have the result of it. He called it sum of the transforming principle. Clear? So everyone, every line is important. So, by this experiment, we were clear that biochemical nature of genetic material was not defined from his experiment. Right? So, it was not happening, right? Even though, right, he said that there's some transformation, but of course, we cannot conclude that what is the nature of the genetic material. So, of course, we saw various other new experiments happening. So, we'll talk about them. So, everyone, are we clear with the first topic? Quick thumbs up, jaldi se. See, I really want to see you interacting with me. Right? Please do stay with us. हाँ, इसीलिए Griffith transformation principle है. Very good. We call it that also. The first thing is clear. Now we'll be moving to the next part, right? Which is very much connected to this particular experiment that the Griffith did. What we have over here? We have three main important scientists. We have Avery, we have McLeod, and we have McCarthy. Okay, and in the book it's written at all. So this is the main scientist, and all of these were working along with him. So we have three main scientists. Please remember their full name also. It can be asked in the examination. So please have a understanding of that. Now what happened was very, very interesting. So when the Griffith experiment happened, the conclusion came. Now scientists were in a half mind, right? They cannot trust. Of course, they don't have a conclusion that what was the result of this experiment. So they were like, no, we'll do some more thing. So here we have three scientists. The full names are over here, everyone, and please make sure you remember them. From the year 1933 to 1944, we saw these scientists performing various experiments to identify the biochemical nature of the transforming principle. Everyone, are we clear? Yes, this is super important. They have to know, they have to find out with what is the nature of the biochemical that is present, right? There. So, of course, that was the, the initiation of the experiment that they have to do. So, उन्होंने क्या किया? We'll see now. So, of course, they also took the heat kill S strain. They made the colonies of the S strain, which is the Streptococcus pneumonia. Then, of course, they isolated the various different parts from it. What are the things they isolated? They isolated the proteins, lipids, RNA and DNA from the cells. So, first of all, what they did, they cultured it. We had the S strain. Then they took the S strain, and from it, they have removed the proteins, lipids. They actually isolated the proteins, lipids, RNA and the DNA. Everyone, are we clear with this? Itna hum sabko clear hai? Good. Now, then they performed the various experiments in the test tube. So, what are the things that they did? So, over here we have the heat killed S strain, and of course we have these test tubes. We have the DNA, we have the protein, RNA, lipids, and of course we have the extract of the S strain also. Clear? Now what they did, and of course they were actually using the R strain also to see that what is actually transforming it. See, for example, if you and me are scientists, if someone tells us, okay, yeah, this is something that I did, do you think you'll be able to believe? No, right? We will be repeating the same steps again to find out whether what the scientist did was correct, or if there are any gaps, we will try to find the answers for it. In this particular case, these scientists are looking for the biochemical nature of the transforming principle. So then they followed the exact same steps that was followed by the Griffith, right? And now, to make sure to find the correct answer, there is an addition of very important thing that they did. Anyone in the class can tell me what they added? Yes? Anyone? What are the things that they added? Yes, they added a very, very important things. Starting with E, right? They added different types of enzymes. Very good, everyone. Right? What they added? They added the different types of enzymes. Now, these enzymes will be able to help in breaking that specific part. And then if it's not present there, we can actually find what is the reason, or what is a real biochemical nature, or what is the material that we are looking for. So everyone, are we clear with this? All of you, are we clear with this, right? What he did, not what he did, what the set of the scientists were doing. Yes, very good, absolutely correct. So they added the enzymes. Now what are the enzymes they added? They added the protease to digest the proteins, RNase to digest the RNA, lipase, right? We have the lipase also, and of course we have the DNAs also. So let's see, everyone, what all happened. So we know that the heat killed S strain extracted the DNA, RNA, lipids, protein, and now what they did? They saw that if they are adding the enzymes, right? Here they added no enzyme. It's a kind of a control thing that you are using. Then they added the RNAs. RNA को RNA के में उन्होंने RNase enzyme डाला. तो क्या होगा? Do you think that RNA will be joined together or RNA टूट जाएगा? क्या हुआ? When they added the RNase enzyme, we saw that the RNA actually got inactive. Basically, it's got broken down. So what will happen? It will not be able to help in the transformation. So हमारा क्या हुआ? कोई भी transformation नहीं हुआ. Yes? Clear everyone? Yes, it will be able to digest it, right? Lipase डाला तो of course we have. Protease डाला तो भी we saw no, nothing happening, right? But जैसे ही DNase डाला, we saw the happening, right? हमें clearly दिखा कि जैसे ही DNase डाला, right? DNA ने क्या किया? DNA को कट कर दिया. DNA जब टूट गया तो अब जो हमारा R strain है वो S में कन्वर्ट नहीं हो पाएगा. That is the reason we see no transformation. Whereas in a case of RNA, protein, right, and lipids, we saw the transformation. That means that when RNase was added, it could not break the genetic material. And hence, the R strain got transformed to S strain. We saw the transformation happening. Same thing happened with the case of lipase, and then the same thing happened with the case of proteinase also. So everyone, are we clear? We saw the transformation and we didn't see the transformation. Everyone clear? Jaldi se all of you give me a quick thumbs up. Clear hai? Thoda sa difficult topic everyone, but please don't lose the hope. So let's quickly revise it. Right? I'm just making it over here for all of us to understand. What we have heat killed. Let me write in this way. Right? We have heat killed S strain. Right? We have R strain. So what they did, they added. So we have DNase, RNase, proteinase and lipase. ठीक है everyone, चलिए हम दोबारा से फटाफट से पढ़ते हैं. All of you, please make sure you stay with me, right? And please make sure if you have any doubts, please do ask me. Okay? Please, if you have any doubts, please do ask me. So we are talking about the experiment done by the Avery, McLeod, McCarthy, right? What they did, they actually started the search and they want to know the biochemical nature of the transforming principle. जो पुराना एक्सपेरिमेंट किया था ग्रिफिथ ने, उसके उनको पता करना था कि बायोकेमिकल नेचर क्या है, जो जेनेटिक मटेरियल उसका क्या एक्चुअली नेचर है. उस टाइम पे रिसर्च पहले ही हो रखी थी, उन्हें कुछ आइडिया था कि प्रोटीन इंपॉर्टेंट है, आरएनए इंपॉर्टेंट है, डीएनए इंपॉर्टेंट है और लाइपेज भी इंपॉर्टेंट है, लिपिड्स भी इंपॉर्टेंट है. तो उन्होंने क्या किया, सेल को पहले तो ग्रो किया, कल्चर किया और उसमें से सेल्स के अंदर में से डीएनए, आरएनए, प्रोटीन और लिपिड्स निकाल लिए. फिर उसको टेस्ट ट्यूब में डाला. अब टेस्ट ट्यूब में डालने के बाद उन्होंने क्या किया, एंजाइम ऐड करना शुरू किया. क्लियर एवरीवन? जब एंजाइम ऐड किया तो उन्होंने क्या देखा, जब लाइपेज एंजाइम डाला तो उन्होंने देखा कि एस स्ट्रेन ने आर स्ट्रेन को एक्टिव कर दिया है और वायरुलेंट कर दिया है. क्लियर? सो उन्होंने क्या देखा, वी सॉ द ट्रांसफॉर्मेशन हैपनिंग. क्लियर एवरीवन? हमने देखा कि अभी भी ट्रांसफॉर्मेशन हो रही है, ट्रांसफॉर्मेशन रुकी नहीं. सेम हैपेंड विद द प्रोटीएज व्हेन दे एडेड द एस एंड द आर स्ट्रेन. एस हीट किल स्ट्रेन विद आर, अगेन दे सॉ द ट्रांसफॉर्मेशन हैपनिंग. क्लियर? कि एस स्ट्रेन ने आर स्ट्रेन को फिर से दोबारा ट्रांसफॉर्म कर दिया है. तो उन्हें कंफर्म हो गया कि लाइपेज का कोई रोल नहीं है, प्रोटीन का भी कोई रोल नहीं है. एंड लास्ट दे ट्राइड विद आरएनए. चलो आरएनए के साथ में देखते हैं. आरएनए के साथ में भी व्हाट वी सॉ के ट्रांसफॉर्मेशन हो गई. राइट? वी सॉ द ट्रांसफॉर्मेशन. एंड लास्ट में व्हाट वी हैड द डीएनए. जब डीएनए को हमने तोड़ दिया तो हमने क्या देखा, नो ट्रांसफॉर्मेशन. तब उन्होंने बोला कि डीएनए इज द की. राइट? डीएनए इज द जेनेटिक मटेरियल. क्योंकि अगर डीएनए नहीं है, डीएनए को अगर हमने तोड़ दिया है विद द हेल्प ऑफ द एंजाइम्स, तो इट इज नॉट बीइंग एबल टू कंट्रीब्यूट इन द ट्रांसफॉर्मेशन. एवरीवन, आर वी क्लियर? यस, ऑल ऑफ यू, आर वी क्लियर विद इट? Yes, very good everyone. So very good. So are we clear with this? Awesome. चलिए, okay. You can take a screenshot of this. Yes, right? Please, everyone, please do take a screenshot of these. So we're done with the second experiment, everyone. Pat on our back. We're doing super awesome. Now, it's a question time, everyone. We'll check our knowledge with the one question. Here we have, in the biochemical characterization of the transforming principle, it was found that process of the transformation is not affected by which of the following enzymes? Which of these following enzymes actually could not affect the transformation process? Right? What options we have? We have A, which is the the enzymes that we have is A is DNase, B is RNase, peptidase, we have lipase. Very good. Option C ko lock kare. Very good, everyone. So of course these three. Awesome. Chaliye. Very good, everyone. So with this, we are done with this topic, and here we have the NCERT ka snip. Let's quickly go through it. So of course, we have to remember the names and the year. From 1933 to 1944, we have three scientists, right? We have Oswald Avery, Colin MacLeod, and McCarthy, right? Three important scientists, right? And they thought that it could be the protein, genetic material could be the protein. Proteins को बहुत ज़्यादा hype मिलती थी. Of course, they worked on to find the biochemical nature of the transforming principle in the Griffith experiment. They purified the biochemicals like protein, DNA, RNAs, etc. from the heat-killed S cell to see which one can transform the live R cell into the S. And they discovered that only DNA can actually transform S bacteria to the R bacteria to the S. Clear? Yes? Hello, yes. Hello, hello. So good to see you, all of you here. Me too, Yash. Okay, everyone, and what he discovered, they discovered that the protein digestive enzyme protease, the RNA digesting enzyme RNase did not affect any transformation. So they concluded that the RNA and DNA is not the genetic material, whereas digestion with the DNAs actually affected the transformation. It stopped the transformation, right? Did affect the transformation, suggesting that DNA caused the transformation. So everyone, are we clear? With this experiment, they were able to conclude that DNA is a genetic material. All of you, are we clear? Yes, it was an indication that DNA is the hereditary material. But not all the biologists will confirm, convinced. But at least we have something with us. Clear, everyone? Chaliye. Yes. So everyone, let's have laddus now. Yeah, right? We are having laddus because we now have an hint कि हमारा genetic material क्या है. At least कुछ scientists को भरोसा हो गया था कि हमारा genetic material क्या है, DNA. So everyone, here we are having the laddus for our discovery. Clear? Yes, yes. Everyone, quick thumbs up. Are we clear with this? Are we clear with this? Jaldi se. And everyone, please make sure, if you're new here, do take a moment, stay with us till the end. And if you're new here, please make sure you hit the like button. And पुराने बच्चे, जल्दी से वीडियो को लाइक करो. प्यार आपका मिलता रहना चाहिए. This is your way of showing, ma'am. We are with you. चलिए, awesome. Yes, Tanu. Yes, Yash. Mayuk, Kuldeep, thank you. चलिए. So everyone, here we are done with this, and this is what we have. I'll not be reading this. It's from our textbook. Yes. Fruits from my side. Thank you so much. Okay. Take a screenshot, everyone, if you want to. It's there in our textbook also. Laddus are my favorite, right? Same here. Chaliye. After this session gets over, right, we'll have some more sweets. Okay, everyone. Now it's a puzzle time for all of us. Very interesting puzzle I have. Or I would say a very easy puzzle. Can you identify this word? Jaldi se bataiye. Can you identify this word? So, when a virus infects a bacteria, the virus is termed as what? We know what we call it as, right? When a virus is infecting a bacteria, what this structure is called as? Or what the virus we call it as? Very good. Easy peasy. It's the bacteriophages. Bacteriophage, right? We know that bacteriophage is a type, of course, it's a virus which actually infects the bacteria. And what they have a protein capsid. The outer structure is made up of the protein, and they have genetic as the genetic material, DNA as the genetic material, which is there inside the core. Yes? So now everyone, we will be understanding that why we are discussing bacteriophage, right? Why suddenly we are discussing about the bacteriophage. So these tiny little organisms, right, we can't call them the living organisms also. So these bacteriophages phages played a very important role in discovering the genetic material, everyone. So are you ready for the experiment? Yes, are you ready for the experiment? Yes. Interesting everyone. Are we ready? Pawar, yes. I think it could be that you're coming here for after a long time. Yes. Welcome to the class. Okay. Here everyone goes to the last experiment that we have, and it's a very easy and super interesting experiment. सारे देखो सारे experiment में से questions आते हैं exam में. ठीक है, तो सारे experiments हमारे लिए special हैं, एकदम दिल के एकदम करीब है सारे experiments. So जो एक last experiment है वो है Hershey and Chase. वो Hershey syrup वाला हम नहीं बोलेंगे उसको, but yeah, Hershey and Chase experiment, right? Of course we have Martha Chase and we have Alfred Hershey. Yes, these two are the important names that we will be remembering, and they actually did this experiment very close to the discovering of the structure of the DNA. So of course the year in which they did the experiment is 1952, and we know that Watson and Crick gave the 3D model of the DNA, the double helical structure, in the year 1953. So it's very close by, right? So let's understand what they did. These two scientists were working, right, and they were planning to at least prove that DNA is a genetic material, right? They have a hint कि हाँ बाकी लोगों ने support किया, but they really want to make sure that this is the genetic material. So what they did, they performed the experiment with T2 bacteriophages along with E. coli. Two important organisms name that we have in this particular experiment, everyone. Please make sure you remember that. Right? So we have T2 bacteriophages and we have the E. coli. And this experiment gave unequivocal proof of the DNA. Right? Unequivocal. मतलब इसके बाद में there is no गुंजाइश, कोई doubt नहीं होना चाहिए कि DNA हमारा genetic material नहीं है. So this experiment is really very important. Exam में पूछा जा सकता है कि which experiment gave कि भैया अब genetic DNA ही हमारा genetic material है, ये experiment, right? We'll remember about this experiment, Hershey and Chase experiment. Clear? उन्होंने क्या किया? Of course they have the bacteriophage जो infect करता है bacteria को, and of course we know that it has a protein capsid and they have the genetic material. Now, very interesting everyone, right? We know that DNA में क्या होता है, and we know that proteins में what we have. Why they opted for the protein and for the DNA? Hello, Cyan, hello. Welcome to the class. Everyone, right? ऐसे cute सा बना सकते हैं ऐसे. Okay? So what he did, right, what these two scientists did, that they took the DNA of course of the protein. को doubt बस इसमें था कि DNA या protein है ना? They're very clear about the other things, but they still have some doubt. So they thought कि with the bacteriophage we'll be able to prove it, because in bacteriophage we have the capsid, which is made up of protein and of course the genetic material which is there for the DNA. So what these two scientists did was that labeling these two different things with the different isotope, radioactive isotope. Yes, what they use the radioactive isotope. Clear? Yes? So, phosphorus and sulfur. Right? We have phosphorus and we have sulfur. Can anyone tell me the numbers? Which? 32. Very good. And we have 35. So, we know that sulfur is present in amino acid. Right? Sulfur is present in the amino acid. Yes or no? Jaldi bataiye. We know that sulfur is present where? It's not there in the DNA. DNA का component पता है हमें. DNA has 3 important components. What we have? Nitrogenous bases, sugar and phosphate. So what they did, they took 2 radioactive elements, right, that we have. We have the phosphorus and we have the sulfur. So we know that sulfur will be present in the protein and phosphorus will be present in the DNA. So what they did? They labeled the proteins with the sulfur and the DNA with the phosphorus. क्लियर? उन्होंने जो बाहर जो कैप्सिड था ना, उसके ऊपर क्या रख दिया? रेडियो एक्टिव सल्फर, ताकि वो चमके. क्लियर? तो जो कैप्सिड हो गया हमारा, वो किसका हो गया? सल्फर का. जेनेटिक मटेरियल अंदर किसका हो गया? फॉस्फोरस का. क्लियर एवरीवन? सी चमकना याद रखेंगे हम. एक्सपेरिमेंट्स में जब ऐसा करते हैं ना, तो उनको लेबल कर देते ताकि व्हेन दे आर ऑब्जर्विंग इट ना, दे विल बी एबल टू आइडेंटिफाई, बिकॉज इट विल बी स्टैंडिंग अलग से फ्रॉम द नॉर्मल. So we have 2 things. I hope that all of you are clear with this. Sulfur, phosphorus, absolutely correct. P32, S35. Clear है ये? है ना? तो यहाँ तक हम लोग clear हो गए. The same thing will take more forward. Clear? So we know that DNA is rich with the phosphorus, whereas sulfur is absent in DNA. Protein is rich with the sulfurous and phosphorus is absent. And hence what they use the P32 and S35, right? They added into the bacteriophages and then they made the bacteriophages infecting the E. coli. अब हम यहाँ पे आगे चलेंगे, we'll come back to this. So what they did, of course, ये first method हो गया. This is the first step, infection, right? We have, this is the radioactive sulfur 35 and over here we have phosphorus 32. उन्होंने ग्रो किया उस मीडियम में, और उस मीडियम में ग्रो करने के बाद में ऑफ कोर्स इस तरीके का हो गया. द कैप्सूल नाउ इज रेडियो एक्टिव, राइट, एंड ओवर हियर द डीएनए इज रेडियो एक्टिव. क्लियर? ये क्लियर हुआ ना? दिस इज वेरी इजी पीजी स्टेप. नाउ कम्स द इंपॉर्टेंट स्टेप एंड नाउ कम्स द इन्फेक्शन पार्ट. सो दे टुक द बैक्टीरियोफेजेस, राइट, एंड बोला कि चलो अटैक करते हैं ई कोलाई पे. सो ऑफ कोर्स दे were attacked. They of course bacteriophage attack करेंगे, इन्फेक्ट करेंगे बैक्टीरिया को. तो बैक्टीरियोफेजेस एक्चुअली बैक्टीरिया के सेल वॉल बाहर के ऊपर आके चिपक जाते हैं. एंड आई एम श्योर आपने सिरिंज देखा है. आई एम श्योर ऑल ऑफ यू हैव सीन अ सिरिंज, राइट? व्हेन डॉक्टर गिव यू इंजेक्शन और वैक्सीनेशन, आई एम श्योर, मतलब वी ऑल सीज दैट के जैसे ही द डॉक्टर विल बी प्रेसिंग, वी सी दैट देयर इज सम मूवमेंट. सिमिलरली ऐसे ही कुछ होता है. जो बैक्टीरियोफेज है, जो अपना जेनेटिक मटेरियल है, वो बैक्टीरिया के अंदर ट्रांसफॉर्म कर देता है, ट्रांसफर कर देता है. क्या कर रहा है? ट्रांसफर कर रहा है. क्लियर? सो बैक्टीरियोफेज जो जेनेटिक मटेरियल है उसका, वो उसने सेल के अंदर, ई कोलाई के अंदर ट्रांसफर कर दिया. व्हाट इज द फर्स्ट स्टेप वी कॉलिंग एज? वी कॉल दैट स्टेप एज अ इन्फेक्शन. क्या किया बैक्टीरियोफेज ने? E. coli को infect कर दिया. In simple terms, E. coli got affected by the bacteriophage as the genetic material is inside the E. coli. Ideally, this is the first step. अब ideally क्या होगा कि अब infection हो गया, we don't need these bacteriophages. फालतू में they will be like those, you know, ऐसे चुपके रहेंगे E. coli पे. We don't need it. So with the step of blending, मिक्सी चलती है ना, उस तरीके से blending करके we will be removing the bacteriophage from the surface of the E. coli. E. coli के surface से हमने बैक्टीरियोफेजेस को हटा दिया. बोला बाय बाय, अब हमें आपकी जरूरत नहीं है. Once we will be removing the bacteriophage from the E. coli, what is the important step? Centrifugation. But before that, we know that once the virus will be releasing their genetic material inside the E. coli will think it's my own genetic material and it will start to replicate. Everyone, are we clear with this? Yes, all of you? Clear? I can see lots of hearts coming. Everyone, are we clear? Clear, everyone? Kuch reactions though? Okay, I can see few reactions coming over here. Very good. Yes, thumbs up everyone. Clear hai? Very good. Chaliye. So we are clear that कि what the bacteriophage will do, they will release the genetic material inside the E. coli, and E. coli will think that this is my genetic material, and it will start replicating it, right? It will make multiple copies of it, and hence what will happen? We'll have more and more of bacteriophages, and that's how the infection happens, right? That's how we know it happens with the virus in a case of virus. So something similarly will be happening over here. Now once that is done, we will have the third and the final step, that is the centrifugation. What is centrifugation? गोल 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 बहुत हाई स्पीड पे घुमाना in a way that we will be able to separate the materials out. Centrifugation works on a principle that the heavier materials or the heavy molecules will come at the bottom, whereas the lighter materials will be floating or will be there in the above layer. Yes, all of you? है ना? मिक्सी में, right, अगर आप सोचो, we have a centrifugation machine with us, right? And of course it works on the gradient, and of course if we rotate at a very high speed, what will happen? The heavier molecules, right? The heavier molecules or the particles, if you want to call it in this case, will come down at the bottom, and the lighter one will be there in the ऊपर, in the top. So after the centrifugation क्या हुआ? Centrifugation के बाद में हमें बढ़िया सा रिजल्ट मिला. कोई बता सकता है क्या रिजल्ट मिला होगा? Right? We have these test tubes. Right? यहाँ पे हमारे पास में P32 है और यहाँ पे हमारे पास में S35 है. जल्दी बताओ क्या हुआ होगा? So let's just have, in S of course we know that we have the genetic material. Yes? Clear, genetic material होगा. यहाँ पे भी genetic material होगा. Yes, और लाइटर में क्या होगा? Of course ऊपर क्या है? तो कौन सा radioactive रहेगा? In which of these test tube you will be able to see the radioactive? Very good, very good. That is the P32. Yes? Very good. Everyone, are we clear? P32 में will be able to see the radioactive isotope. मतलब of course यही है. इस test tube में हमें दिख जाएगा. That means that clearly means that कि during the infection, the bacteriophage which has the protein which has the radioactive S, right, sulfur 32, that could not come inside the E. coli, right? जो प्रोटीन का जो कोड था, जो प्रोटीन जो हमारा रेडियो एक्टिव था, वो तो अपने जो इंफॉर्मेशन है वो आगे ई कोलाई को दे ही नहीं पाया, क्योंकि इंफॉर्मेशन कहाँ से आ रही है? इंफॉर्मेशन आ रही है डीएनए से. वेयर एज जहाँ पे हमने P32 डाला है वो डीएनए हमारा रेडियो एक्टिव दिख रहा है हमें यहाँ पे. क्लियर? सो हमें P32 दिख रहा है बिकॉज वो हमने डीएनए में डाला था और डीएनए ही ई कोलाई के अंदर आया, और दैट्स हाउ वी सॉ दी ट्रांसफॉर्मेशन. क्लियर एवरीवन? इसी तरीके से आगे ट्रांसफर हुआ. एवरीवन, आर वी क्लियर विद दिस? जल्दी से बताइए, ऑल ऑफ यू, आर वी क्लियर विद दिस? यस नरोटो, आई विल एक्सप्लेन. यस एनोनिमस, नरोटो, हनामंतु, ऑटो, तनु, वेरी गुड. क्विक थम्स अप एवरीवन, जल्दी से. राइट? सो आफ्टर द एक्सपेरिमेंट वी कैन क्लियरली सी कि we have the ghost particle, the labeled ghost particle over here, and unlabeled DNA. So unlabeled DNA यहाँ पे आ गया, which have no color, right? And over here we have the labeled DNA. So जहाँ पे we have the labeled DNA, जो DNA चमक रहा है, that means that it actually got the information from the DNA, from the bacteriophage. So hence with this particular experiment, the scientists concluded that the DNA is the genetic material. Happy faces everyone. अब जाके साइंटिस्ट को शांति मिली कि हाँ भाई, जो हमारा डीएनए है ना वो ही जेनेटिक मटेरियल है. एक और चीज जो आई थिंक आई जस्ट मिस्ड ओवर हियर, तो यहाँ पे दो चीजें होती हैं. जो नीचे वाली जो मॉलिक्यूल्स जो बॉटम पे रहते हैं उन्हें हम क्या बोलते हैं? वी कॉल देम एज दी पेलेट, एंड जो ऊपर हैं उसे हम बोलते हैं सुपरनेन. ओके, सो यू कैन नोट डाउन दिस बच्चे. चलिए, ऑल ऑफ यू क्लियर? बढ़िया, समझ में आ गया सबको? जल्दी से चमक गया ना? सी वहां पे रेडियो एक्टिव आइसोटोप चमक रहा है. क्या हमें कॉन्सेप्ट चमका? यस, क्या हमें कॉन्सेप्ट चमका? यस, वेरी गुड, वेरी गुड. चमक गया ना हमें कॉन्सेप्ट. सो नाउ वी आर क्लियर कि किस तरीके से साइंटिस्ट हैव प्रूव्ड दैट डीएनए इज द जेनेटिक मटीरियल. Now everyone, question time for us. चलिए, here we have a question for all of us. In the Hershey's and the Chase experiment, radioactive P32 was used to culture bacteriophages, which resulted in radioactive what? Viral DNA, bacterial capsule, viral protein, or protein capsule of bacteriophage. Which one of these is the correct answer? Option A lock करें. It's an easy question. हाँ, ये easy question है. A very quick revision. Very good. That's a viral DNA. चलिए, next question everyone. Next question. The final proof of the DNA as a genetic material came from the experiment of. अभी पढ़ा तो I'm sure you'll be able to give the answer. Yes? From where we got the final proof? Very good. By the experiment done by the Hershey and Chase. Option B is the correct answer. So with this everyone, we are done with the experiments. There's one more छोटा सा topic that we have. See over here. 1952 में this experiment came, right? And of course they did this experiment. We have the bacteriophages. The bacteriophage will attach to the bacteria and its genetic material will enter into the bacterial cell. This bacterial cell treat the viral genetic material as its own and manufacture more viruses. So these two scientists worked on this, right? Whether it was the protein they were looking for, they have to pick one amongst the protein or the DNA, right? What they did, they grew some bacteria on the medium containing radioactive phosphorus and some containing radioactive sulfur. Viruses grown in the presence of phosphorus contain radioactive DNA, whereas the virus that are grown on the radioactive sulfur have the proteins. Everyone clear? They have the radioactive proteins, but they don't have the radioactive DNA. Uski baad mein what they did? Then of course they followed the experiment. Radioactive phages were allowed to attach to E. coli. Then the infection proceeds. Viral coat was removed by the agitating them in the blender. And then what we saw that the virus particle was separated from the bacteria by spinning them in the centrifuge. Clear? Then what we saw that bacteria was infected with virus that has radioactive DNA were radioactive, indicating that the DNA is a genetic material which is passed from virus to the bacteria. And after this, bacteria that were infected with the virus with the radioactive protein were not radioactive. So this indicated that the proteins have no role in as a genetic material, whereas DNA is therefore the genetic material. Clear everyone? Clear all of you? So this is super important. NCERT पढ़ ली हमने समझा और NCERT भी पढ़ ली साथ में. सही चीज है. Everyone, this is super important. आप please make sure to remember this diagram. हमें बार बार ये डायग्राम देखना है. इसमें से बहुत कुछ आ सकता है हमारे नीट एग्जामिनेशन में. जितने भी डायग्राम है हमारी एनसीईआरटी में सुपर इंपॉर्टेंट. वी हैव इन्फेक्शन, ब्लेंडिंग एंड द सेंट्रीफ्यूगेशन. प्लीज मेक श्योर यू रिमेंबर दीज डायग्राम्स एंड ऑफ कोर्स वी नो दैट दीज आर द easiest way for us to remember. Done? चलिए, all of you, are you clear with this? A quick thumbs up जल्दी से. I can see that we have so many of you here. Everyone, please make sure you hit the like button and फटाफट से सब लोग कुछ स्माइलीज बना दो. चलो जल्दी से स्माइली बना दो. I know that थोड़े से थक गए होगे, right? The class is almost 1 hour done. Just छोटा सा topic है, small topic that we have. We'll finish it up, and then of course next week we'll start with the DNA replication. Interesting. I can see hearts coming. We have some emojis coming. Very good. Interesting. We have tomatoes also. Nice. हाँ, टमाटर अभी थोड़े से महंगे भी हैं तो ठीक है, ये भी भेज दो. चलिए, so we are done with the 3 important experiments everyone, and we have seen few questions based upon them. Now let's move ahead to the last, the smallest part that we have, and after this we will be finishing our session. We already had a discussion about it. But let's quickly put it on the slides and on the sheet so that we have the greatest memory of this. That why we prefer DNA as a genetic material, why there's a lot of comparison between these two. So let's see that why we prefer DNA as a genetic material and not the RNA. So we know that DNA is now the genetic material after the so many experiments, right? And we also know that some of the viruses have RNA as a genetic material. We have the example like tobacco mosaic virus, QB, bacteriophage, right? So this is again from your textbook. Now in your textbook there is one line that I have mentioned, okay, both of them can act as a genetic material, right? And there are some criterias for a molecule to be a genetic material. Now, what are the criterias? First, they should be able to replicate. They should be able to replicate themselves. They should be chemically stable and structurally stable. They should have a small scope of mutation also, slow changes, so that evolution process can happen. And last and most important, that they should follow the Mendelian characteristics features. Dominate, recessive, etc. Everyone, are we clear? So these are the criteria. See, those of you who are in class 12th, this question can come for 2 marks. 12th board student, this question can come for 2 marks. Now, so of course we know, if we compare this everyone, we know that both of these DNA and the RNA can generate themselves, right? They can easily replicate. We know that both of them have a property of the replication. They have the complementary base pairs, they have the base pairs. So of course they'll be able to replicate themselves. So that is one thing. Then of course, they provide the scope of slow changes also. Definitely they do that, right? They have the scope of changes so that the evolution process can go on. But which of these is more stable? DNA or RNA? We know that DNA is more stable as compared to the RNA, right? Both chemically and because of the structure also. Can anyone in the chat, everyone quickly tell me that why RNA is not stable? We have discussed about the structure of the RNA, right? That why it is not that stable and why DNA is more stable. Yes, everyone. We're talking about the structure. We know that we have the ribose sugar. We made the structure. We remember we made the structure of it. We remember there's a presence of the OH group. Absolutely correct. Now this OH group is not present in the DNA, but this is only present in the RNA, and because of this OH group, it's very active. It is always ready to form a reaction, right? It's ready for the reaction. Yes, I'm ready for the reaction, right? And it's catalytic in nature, catalytic in nature. It wants to get involved in the reactions. So this is not chemically stable, and of course structure-wise also it's not that stable, and hence we can clearly say that RNA cannot be the genetic material, right? And of course we can mention about the point of the uracil and thymine. Yes. So we know that again we have the replacement. In RNA we have the uracil and in DNA we have the thymine. Again, okay. Then the last point is that they should able to express themselves in a form of the Mendelian character, and we know that, yeah, pretty much they have. Okay. So everyone, are we clear with this? Are we clear with this? Very good. Yes, uracil is more reactive. It's definitely more reactive. Okay. Yes. Manvendra, hello. Chaliye. So with this everyone, we are done, and the last topic that we have, the RNA word, right? We know that RNA is a very first genetic material that we have. When we always talk about the genetic material, we always go back to the RNA. RNA पहला genetic material है. क्यों है? Because we have the enough evidence, because it's there in various life processes. हमारी body में nucleus के अंदर, right, we have nucleolus, and वहाँ पे कुछ ऐसा बन रहा है जो RNA में है, right? And it actually help. Yes, it actually help. So we know that it is there in a various essential life processes like metabolism, translation, etc. This RNA uses genetic material as a catalyst, as we were discussing now only. ये हर जगह पे use किया जाता है. Yes. So we know that there are biochemical reactions happening in the living systems. There are that really requires the RNA and not the protein enzymes, right? And RNA being a catalyst is more reactive and more unstable. Therefore, there has to be some modification that we see in the DNA and it becomes more stable. So, DNA में कुछ modifications हुई हैं, evolve हुआ है and it's more stable. Clear? Clear everyone? Everyone, are we clear? Yes, so then we know that the DNA being the double-stranded and have the complementary strands further resist the change and evolving process of the repair. Very good. Awesome. So are we clear with this, everyone? Good. Yes. Okay, interesting, everyone. So are we clear with all of these questions? And are we clear with all of these concepts that we have studied? Good, very good, very good. Yes. Crystal clear, very good, very good. Chaliye. So with this everyone, we are done with our class. As a homework question for all of you. There are 2 homework questions. Please write the answer in the comment section. So here we have question number 1 and here we have question number 2. I will be waiting for your answer. Okay? So I would love to see everyone. Please do write in the comment section and write the answers. Yes, so with this everyone, we are done with our class. Thank you so much everyone for staying with us till the end. Right? In the upcoming weeks, we'll be discussing about the DNA replication, transcription, right? And we have interesting classes planned for you. So please make sure you watch all of these sessions that are happening. We have physics taken by Mrinal sir, we have zoology taken by Sneha ma'am, chemistry taken by Ashish sir. So please make sure you join all the classes, right? And everyone, please make sure, right, to subscribe to the channel and hit the like button for the video. Yes, interesting everyone. On that note, everyone, I'll say bye-bye. Do take care of yourself, everyone. Please read the NCERT once again. If you have any doubt, you can definitely write in the comment section and ask doubts, right? It will be perfectly fine. I'll be replying to your doubts. Yes. So, everyone, do take care of yourself. Lots of love to each one of you. Do keep yourself super healthy, keep yourself hydrated, eat good food, and keep on learning with BYJU'S. Lots of love to each one of you. Bye-bye, everyone. Good night. See you soon. Bye-bye.